Hello. Yeah, I am waiting for a few more students to join. After that, I will be starting with the live session. Sorry, I did not get you.
कॅमेरा इथं चालू होते आणि मी इथं बंद केलं तर माझं ऑफ होते. याचा अर्थ माझं
Kindly mute yourself. That will actually help.
hello sir,
Yes.
are you listening me?
Yes, yes.
I'm first time using this properly, so it sometimes problem.
Sorry, I did not get you.
Google Meet this app I am first time using. So
Yeah,
I
yeah.
am
This is actually the first. You can say the first live session that I'm taking.
uh,
So, fine. Okay, so yeah, I think we can start, and after that, if someone is coming, we can join. They can join later. Okay, so I welcome you all to today's week one live session on the course Experimental Biochemistry, which is taught by Professor Shagota Dasgupta and Professor Shomode of IIT Kharagpur. And I am Srijan Sen, and today I would be taking the week one live session class. Okay, so uh, and in the beginning I would be discussing with you people the lecture slides in brief, and then I would be discussing the assignments. Okay, the previous year assignments or new questions or how to solve the questions, I would be doing that. So uh, in the middle, if you have any questions, you can switch on your mic and ask me, or you can even ask me in the text, uh, in the chat box. That is all fine. And so in the first I would be discussing with in brief the lecture slides, and in the next half um, I would be discussing the assignment. Okay, so this live session will happen every Monday from 6 p.m. to 8 p.m. So all are welcome to join the live session to solve your doubts, and if you have any queries and all, because this is a one is to one session. Okay, so even you can ask your friends to just register for this course and join the class. Okay, so I am sharing my slides with you all. Yes, so is my slide visible to you all? Kindly someone please confirm. And am I audible or not?
you are audible, but your slides are not showing properly.
My screen is visible, right?
Uh,
Right.
now getting sir.
Yes, yes, fine. Okay.
Again,
This is the week one live session. So in this class, we would be mostly covering the concepts on accuracy and precision. So in today's class, we would be mostly discussing the concepts of accuracy and precision. Then we would be talking about acids and bases, followed by some important informations that we need to keep in mind about pH and Henderson-Haselbach's equation, and then we would be speaking about buffers. So let us begin one by one. So firstly, when we speak about accuracy and precision, uh, these two terms are the most important factors of measurement. To talk about measurement, these are the two important things that we always need to speak of. Otherwise, the thing remains incomplete. So, what is precision? So, it is basically the closeness of the um, two or more measurements to each other. So, how close your measurements are, that is precision, and it is independent of accuracy. Okay, so it does not depend on anything. If you have a particular, say, value and you repeat the experiment twice or thrice and you get that same value, it may not be accurate, but you can call the value to be precise. And when you talk about precision, you separate it or divide it into two categories. One is repeatability and the other is reproducibility. So what are these two terms? Repeatability, so it is the variation that arises when you keep all the conditions and the environment identical, and you repeat the experiment or you repeat the measurements that for a short period of time. Say you repeat it twice or thrice or four times. Okay, that's not for a long time. Now reproducibility on the other hand is for long term period, and it arises by using the same measurement process but in different instruments, or you operate it differently, but you get that same value, and this is a long term for long term periods. So precision is the closeness of the points or the closeness of the measurements independent of accuracy, and it should be repeatable and reproducible. Okay, now when we speak of accuracy, on the other hand, it is the ability of the instrument to measure it more correctly. Okay, so now we know that when we are pipetting solutions, if you are aware of pipetting solutions, or when we use a pipette, when we set it to 1 ml, that is accurate. This measurement is set to be accurate. Okay, so when I'm picking up pipetting 1 ml, it will be actually 1 ml. So we know that. So that is accuracy. So it is the closeness of the measured value to a standard or a true value. So what is a true value? A value that is known to us, and we are matching it with our with the value that we have got. And this reduces the error of the calculation. So precision and accuracy. Now, how do we know that whether the measurement is accurate or precise? Now, if we see all of these four circles, when we see all of these four circles, we can see the four situations. So the first situation is a scattered point around the central, around the center of the particular sphere or circle. Now the points are scattered more or less around it. So you can tell when you do the average of it, you can tell that the points are accurate, but they are not precise because they are not at a particular value, but they are more or less scattered around the accurate or a known value. Now, when you go to the next one and you see that the points are scattered everywhere, so that means it is neither accurate nor it's precise because it's not populated around the center of the sphere. This situation is very deviating from the center point or a true value, but it is aggregated or populated at a particular part of that uh, circle. So you can call that this is precise but not accurate. And in the last scenario, it is populated at the center as well as the points are more or less similar. So you can call this precise as well as accurate. Okay, so these are the four different scenarios that can arise. The first one being accurate but not precise, accurate and precise, not accurate, not precise, and precise but not accurate. Okay, so and this can be plotted in a particular x and y axis. So let us take an example that the volume of a liquid is 20.4 ml. Which of the following set represents the value with a good accuracy? So we know the true value is 20.4 ml. So we need to just find out which of the values is more or less very close to the unknown value. So so you people can try doing it and tell me the answer as to what it will come. So the volume is given, and all of these are the values received. So which of the following set represents a value with a good accuracy? Anyone trying? Okay, so what you need to do is you have to calculate the average of these four values, and you will get the value which is very close to 20.4 will be the correct answer. Now, if you see from the four options that are given, you would be seeing that the last option is close to this particular. When you do the average of the last option, you would get the more or less an accurate value. So next, let us discuss about the uh, difference between accuracy and precision. So accuracy is the closeness with the true value of the quantity that we are measuring, and precision is about the reproducibility of the measurement. And the measurement can be accurate but not necessarily be precise, but the measurement can be precise but not necessarily it is accurate. Okay, so so the first one being it's the closeness to the true value. The second one is speaking about the about reproducibility. Here the measurement can be accurate but not uh, precise, and here we have the measurement can be precise but not accurate. Okay, and for accuracy it is determined with a single measurement, and it needs several measurements to be determined, because when we speak of precision, how do we tell? Now you measure something and you see that it is very close to the true value, that is being accurate. Okay, but for precision you have to get points. When you get a point, then second point has to be close to that first point. When you do the experiment the third time, it has to be very close to either of the first or the second point. So it needs several measurements to be
Hello.
yeah.
Good afternoon, sir. So Gayatri Jaiswal this side. Actually, I have a question.
Yes.
Hello, am I audible, sir?
Yes, yes, tell.
Sir, actually, I don't know anything about the course. Can you please explain? Just I'll take 2 minutes.
Okay, Sure. So great.
Sir, can you talk in Hindi, please, if you're comfortable?
No, actually, this is a recorded class, so I have to stick to one language, and that is English.
Okay, that's fine. So can you please explain me about the course? I really don't know anything about that where to attend the classes or anything else.
Okay, so no means where to attend the classes means you have come to the live session because you've got the mail, right? If
Okay,
not,
so
if
only we can get an information through mail only, na? There is no app or there is no message or there is no WhatsApp, na?
you have to do the slides. Means sorry, you have to
Actually,
register
I
for the course.
purchased the I I purchased the session this course tomorrow, yesterday.
Yes, that is fine. I understand. Okay.
Hmm.
So
Okay,
I am telling that uh, you have to first register for the course, and you have registered it yesterday, right?
sir, I registered. Yeah, I registered for the certificate also.
Right. So now let
Yes,
me
sir.
tell you. So every Monday, and
Yes, sir.
I think your classes have started from 22nd.
Yes, sir. Yes, sir.
From 22nd to means every week there is going to be a lecture.
Okay, every week, every Monday.
No, yes, because it
Oh,
has started from Monday, so every week
yes,
there
sir.
is going to be a lecture, and that lecture is
Okay,
a
sir.
recorded lecture that will be available in the that Swayam portal.
Okay, sir.
So the
Okay,
lecture
sir.
is
So I
available
get the
only there.
lecture from the. So
Yes,
now onward, I mean to say today, I also eligible to get that lecture, no? I mean to say I can see that lecture.
see, firstly
Yeah.
you have to register for the course. Okay,
Yes, sir. I register.
yeah. Secondly, in that portal you will get everything.
Okay,
Means
sir.
where you registered, you will get the lecture
Yes, sir.
slides, everything. Okay, and that is a
Yes, sir. Yes, sir.
recorded lecture. Okay,
Yes, sir.
get it.
Yes, sir.
Now what you have to do is every week you have to go, you have to listen to the lectures, and you
Yes, sir.
have to solve the assignments.
Yes, sir. Okay. Assignment means the homework, na? Which
Yeah,
you gave
so
us.
because that has a percentage of
Okay.
marks in your
So this
final
is
exam.
it? Yes, sir. Is it compulsory to do the
Yes,
assignment?
yes, otherwise you will
What is
break
the
test.
drawback to not to done the assignment?
The drawbacks is that that percentage of marks won't be added to your final examination, and you have registered for the examination. So already, so it will be like you are giving an exam but won't getting a certificate if you do not cross a certain percentage. If you do not get a certain percentage,
Okay,
so you
so
have to do the assignments.
exam is not sufficient. Now I also have to done my
Yes,
assignment.
so there is a percentage of
Or
percentage that will be added.
what if I only write the exam and not doing the assignments regularly?
Now that is up to you, but it is always suggested or recommended to do the assignments. Now, see, the benefits of doing the assignment and coming for the live session is that those assignment from the assignments there would be questions, there would be similar type
Hmm.
of questions. So if you solve the assignments, it will
Okay,
help you
okay,
to
okay.
do solve the questions in the exam.
So the
Okay.
in assignment, how many questions are there?
I guess there would be about 15 to 20 questions maximum. About 15.
Okay. So I have to solve these questions.
Yes, you have to solve these questions. Okay. Now,
Okay, 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 okay.
what we do, or what I will be doing in my live session
So
is
this is I want to say that actually I am doing a job, so this is not possible for me to do the assignment. Actually, I can attend the class, but this is not. I mean, to say I'm unable to do that assignment. So if I am not done the assignment, then
you
am I not eligible
see,
to do the exam also?
you are doing some job, that is fine, but the assignment can be done anytime.
Yeah,
You can do the
that's true.
assignment on a
And what
Sunday,
if
on a Saturday, on any day.
what if all of my answers? What if all of my answers get wrong
See,
in the assignment?
see. It is not like that. That is why we take the live sessions, okay? Such that you
Okay,
can,
okay, okay.
firstly, I do not know actually after the live session the assignment gets submitted or before the live session, that I do not have an idea about. But we
Okay,
do
sir.
the live session such that you can do the assignments correctly and you can give the exam well. That is why we are taking the live sessions.
Okay, sir. So the live session which held on Monday.
It will be held on
So can
Monday,
I see
6
that
to 8.
video lecture?
Get
Can
you.
I see that video lecture?
Yes, it will be available on YouTube. You can definitely see. But it is always good to come to the class and solve your
Live session.
doubts.
Yes, sir. Yes, sir. No, sir. Now onward, I'm trying to attend the live session, but on Monday, I mean to say, I purchased the
Okay, so on Monday it's
yesterday.
on Monday 6 to 8 and on Tuesday 6 to 8.
Yes, sir.
So these
Two
are the
days.
2 days.
They are class
No,
on
no,
two
means
days.
either of the one you can attend. You can either
Okay,
attend on
either of the
Monday
one.
or on Tuesday.
Okay, okay. Okay, sir. Okay, sir. So this class is for what?
Yeah, so it is, this course is on experimental biochemistry. Now here
No, sir,
if
I'm
we
talking about the today's session.
yeah, today's
You're
session will be
solving
on
the
the, yeah, I would be solving the assignments, means I would be
okay,
discussing the
okay. You're
assignments.
solving the okay, okay.
And
So here is I mean that I am unable to solve the question. So you are there, or any other teachers are there to solve the assignment.
right, okay.
Okay,
So we
sir.
won't be
Okay, sir. Now
discussing
I get
this
it.
year's assignment, but we would be discussing
Okay.
the previous year's assignments.
Yes, sir. Yes, sir. Okay,
Okay,
sir.
you get it? Okay.
Okay,
If you
sir.
have any
Yes,
more
sir. You can
queries,
continue. Thank you very much, sir.
okay, if you have any more
I have
queries, we
lots
will
of
discuss
doubt
after the
because
class. Right.
yes, sir.
You can
Sorry to disturb
continue
you,
with
sir.
queries. Yeah, that's fine. That's fine. It's always good to
You
get
can
the
continue,
doubts
sir.
clarified.
You can continue. Thank you so much, sir.
Okay, so while we stopped, so let us start from this. So we were discussing about the difference between accuracy and the precision. So firstly, accuracy is the closeness to a true value, and precision is how reproducible your values are. Now, a measurement can be accurate, not precise, and on the other hand, the measurement can be precise but not accurate. And accuracy is upon a single measurement. So, when you know the true value and you do a lot of calculations and all, and you get go close to the true value, that is fine. It's an accurate value. But to be precise, it is a particular. So, you do the experiment once and then you repeat it twice, thrice, the same values you have to get a precise value. So you have to repeat the experiment a several number of times. So that is several measurements to be determined, and the accuracy gets affected with a systematic error and precision is affected with a random error. And it's about, accuracy is about the degree of conformity and the precision is about reproducibility. Okay. Now, these are a few examples of accuracy and precision. Now, if the weather temperature reaches 20 degree and it is 20 degree outside, so the measurement is said to be accurate because we know the true value and it is actually the same. Now, if the thermometer continuously registers the same temperature for several days, we call the measurement to be precise. So, if it is 20 degrees for 4 to 5 days or say 6 days, because as I was speaking, repeatability is of a short term and reproducibility is of a long term. So, when you get the same value for a long period of time, you tell that the value is reproducible and it's a precise measurement. Now, if someone is taking the measurement of the mass of a body is 50 grams and you get these values, and on a weighing scale it is precise because it is 47.2 on an average, but it is not accurate because it is deviating from the 50 kg mass. But if the scale reads 49.8, 50.5, and so it is more accurate than the first balance but not very precise because the standard deviation the deviation from the main point is more, but when you take the average of these 4 points, you get close to the 50 kg mass. So that is all. Okay, now we have a question over here, that is a new 5 rupee coin weighs 6 grams. Two new weighing machines were tested with this coin by measuring its weight 5 times and the value was recorded. So for the first weighing machine gave values of 5.97, 5.99, 0.98, 0.96 and 6 grams, and the weighing machine B gave 5.98, 6.01, 6.02, 5.97 and 5.99. Now, if the question is which of the following is true? A is more accurate, B is more precise, A is more precise, B is more accurate, or A is more accurate and precise than B, or A is less accurate and precise than B. So these are the 4 options. First, you people try to solve the question, then I would be telling you the answer. And for giving me answers, you can definitely use the chat box to give me the answer, or you can just speak it out, but it's better to write in the chat box as that it remains. So you try solving it, then I would be discussing. So similar type of question I had also solved previously. So this is just the question in a different type. So I think it's not very tough. You can definitely do it. Every one of you should try to solve these questions. Okay, these might be some questions will be easy, some questions might be tough and tricky, but try to solve the questions in this live session such that if you have any problems, your doubts can get cleared over here. Anyone with an answer? No. Okay, so uh, let me discuss this one. Okay, so the first weighing machine is measuring 5.97, 99, 98, 96 and 6 grams. So when you do the average of these 5 measurements, you get an average of 5.98. Okay, so you get an average of 5.98, and when you do the average of the weighing machine B, you get 6 grams. Okay, the average. But you see that for weighing machine A, the values are more precise, but they are not accurate. But for weighing machine B, the values are accurate but not precise. So A is more precise because the values are more or less on, they are not deviating much from the average value. But for weighing machine B, the values are more or less different, but when you take the average, it is very close to the true value. So that's why B is more accurate and A is more precise. So option B is the correct answer. Okay. So moving on to the next topic, that is on acids and bases. So a free hydrogen ion, it does not actually exist in water. It forms a hydronium ion because of its attraction to the polar water molecules is so strong that the H plus bonds to the water molecule and we get this hydronium ion or H3O plus. Now, water acts as, why are we choosing water? Because water acts as both as an acid as well as a base. So when we take water in an acidic medium, say HCl. So HCl plus water generates hydronium ion and the chloride ion. Now water acts as an acid where when we take it in the presence of a base. So we have ammonia with water, we get the ammonium ion and the hydroxide ion. So the generation of an hydroxide ion gives us a basic solution and the generation of the hydronium ions gives us an acidic solution. And water acts as a base in the presence of an acid and acts as an acid in the presence of a base. So it has dual characteristics. Now, speaking of the conjugate acid-base pairs, so according to Bronsted-Lowry, this conjugate acid-base pair exists that consists of molecules or ions that is related by either the loss of one hydronium ion or the gain of one hydronium ion by the base. So loss of an hydronium ion by an acid and the gain of an hydronium ion by a base. And this um, acid base reaction contains two conjugate acid base pairs because the hydronium ion is transferred to one and is taken up by the other. So if you see that we have hydrogen fluoride with water, where hydrogen fluoride is the acid and so hydrogen fluoride is the acid and water is the base. So against a base we have a conjugate acid, and for the acid we have a conjugate base. So when the hydrogen fluoride loses an hydronium ion, it becomes a conjugate base, and this whereas this ion is taken up by this water molecule and we get the conjugate acid of this particular base. Now, what is the conjugate acid base pair? So you have the HFF to be the conjugate acid base pair and water and the hydronium ion to be the conjugate acid base pairs. So we have two conjugate acid base pairs because for the first case the hydronium ion is getting transferred or the hydrogen ion is getting transferred and it is taken up in the reverse or the forward directions. So this is an acid, it loses an hydrogen ion to get the base, conjugate base, and water as a base is gaining an hydrogen ion and it gives us a conjugate acid. And similar kind of a reaction is also occurring for ammonia with water. Now let us discuss about the dissociation of water. So water is amphoteric, that means it has a dual characteristic that it can act as both as an acid as well as a base. So in a particular reaction of water we have also two conjugate acid base pairs. So when we have two molecules of water we get a hydronium ion and an hydroxide ion, where one water molecule is the acid and we get a conjugate base for it, and the other water molecule is taking up a hydrogen, so it's the base and this is the conjugate acid. So what happens in pure water? There is a forward reaction between the two water molecules that transfers the hydrogen ion from one water molecule to the other. The other molecule acts as an acid by losing the hydrogen ion, and the water molecules gains an hydrogen ion that acts as a base. Now, every time this transfer is occurring between the water molecules, the products that we get are hydronium and the hydroxide ions, which react in reverse direction to form two water molecules. And that's an equilibrium between the water molecules and the formation of the ions. Now, this is very important where we would be discussing about the dissociation expression of water. So from the previous equation, if we try to draw the chemical equilibrium, So that is K equals to your hydronium ion concentration with the hydroxide ion concentration divided by the concentration of the water molecules. So when we write the chemical equilibrium of a water molecule, so we consider that the concentration of the water molecule is negligible. Okay, now as it is negligible, so we can omit this, the concentration of water, and we can write it the K, the chemical equilibrium, as the equilibrium of water, as Kw, or the water dissociation expression. That is Kw equals to the multiplied version of your hydronium ion and the hydroxide ion. And when you repeat the perform these experiments in pure water, that is at 25 degrees centigrade, it is 10 to the power minus 7 molar for each of the concentration, because it's a pure water molecule. So pure water has equal concentration of hydronium and hydroxide ions, that is 10 to the power minus 7 molar, and Kw turns out to be your 10 to the power minus 14. So because it's the multiplication of the two concentration terms. Now to find out the concentration of your hydronium ion, when the hydroxide ion concentration is known, or vice versa, you have to find out the hydroxide ion concentration when the hydronium ion concentration is known. You can just do it in this manner, where you know the value of Kw, that's 10 to the power minus 14, and you can divide that value by the concentration of the hydronium or the hydroxide ion that is given, and you can get the concentration of the other term that is not given to you. Okay, so speaking on the pH scale, so acidic solutions, we call acidic solutions to those solutions that have pH less than 7, that is the hydronium ion concentration is greater than 10 to the power minus 7 molar. The neutral solution has a pH equals to 7. So the concentration of hydronium ion is 10 to the power minus 7 molar. And for a basic solution, the pH is greater than 7. That is, hydronium ion concentration is less than 10 to the power minus 7 molar. On the other hand, if we speak from the perspective of an hydroxide ion, that is for the acidic solution, hydroxide ion greater than 10 to the power minus 7. For neutral solution, hydroxide ion equal to 10 to the power minus 7. And for the basic solution, hydroxide ion concentration greater than 10 to the power minus 7 molar. And these are the different indicators that we use to find out the color of, to find out the pH of a particular solution. This is the pH range and the indicators that we use to find out the acidity of a particular solution. And this is the pH value of the different items that we either consume or we use in our daily lifestyle. Okay, so it is given over here, and the slides would be shared with you. You can check back with them. Okay, so the another important point is to calculate the pH and the pOH of the solutions. So the pH scale is the logarithmic scale that corresponds to the hydronium ion concentration of aqueous solutions. So pH is the negative logarithm to the base 10 of the hydronium ion concentration. That is, pH equals to minus log of hydronium ion concentration. For example, if we have a lemon juice that is an acidic solution with hydronium ion concentration of 10 to the power minus 2 molar, it has a pH of 2. And how can you calculate it? So pH minus log of 10 to the power minus 2 to the base 10 of logarithmic, and you get a pH of 2. So in this manner you can calculate the pH. The pOH scale is similar to that of pH, except here we have the hydronium ion concentration and here we have the hydroxide ion concentration. So we can do in that manner. Also, what we know is that Kw is equal to 10 to the power minus 14. So if we take log on both the sides, okay, so if we take log on minus log to the base 10 on both the sides, we get minus log of Kw is equal to minus log of hydronium ion concentration minus log of hydroxide ion concentration. So minus log of particular concentration equals to pH. So here we have pH plus POH equals to minus log of Kw, and minus log of Kw is equal to 14. So from there we get pH plus POH equals to 14. Okay, so in this manner also you can calculate, when the pH is given you have to calculate the POH, you can get it, or vice versa, you need to find out the POH but the pH is given. So for example, for this example you have the pH is given as 2. So if someone tricks the question in this manner and asks what is the POH, you can do it, 14 minus 2 is 12. It's as simple as that. Like here one example is given. Okay, now in summary, pH equals to minus log of H plus or H3O plus hydronium ion concentration, and POH equals to minus log of hydroxide ion concentration. But the units have to be kept in mind, that is, all the concentrations have to be in molar. Okay, if something is given in millimolar or micromolar or nanomolar, we have to convert it to molar, and then only we get the value of pH or POH. And the product of these two concentrations is 10 to the power minus 14, and from there we get pH plus POH equals to 14. So an acid is a proton donor, and so here AH is the acid, we have its conjugate base, and so a base is also defined as a proton acceptor. So here we have B, and this accepts one proton, so it's the conjugate acid, and B is the base. Simple things. Okay, now let us speak about weak acids and pK. Okay, so we can know the strength or study the strength of an acid by when we determine its dissociation constant, or the Ka. So acids that do not dissociate significantly in water are known as weak acids. So to study the dissociation of an acid, it is Ha. For example, we are taking this equation, Ha equals to H plus plus A minus. So the equilibrium, so the chemical equilibrium would be a concentration of A and H plus divided by the concentration of the acid. Okay, so when so this is how we calculate the dissociation constant. Now when this value is less than 1, your concentration of HA is greater than the other two, the product of the other two concentrations, and HA is not significantly dissociated. So we can call HA to be a weak acid when when the dissociation constant is less than 1. So lesser the value of Ka, weaker will be the acid, and higher the value of Ka, stronger will be the acid, because it is dissociating faster. How can we tell it is dissociating faster? When the product formation is more. So when product formation is more, that means the Ka value is more. More Ka value means stronger acid, less Ka value means weaker acid. Okay, so similar to pH, the value of Ka can be represented as pKa equals to minus log of Ka. Okay, so the larger pK, the weaker acid. And pK is a constant for each conjugate acid and base pair. So most biological compounds are weak acids or weak bases. Now polyprotic acids. So what are polyprotic acids? A particular molecule or a substance that is capable of donating more than one proton. Okay, it's called a polyprotic acid. Like we have the diprotic and the triprotic acids are specific types that donate 2 or 3 protons. So diprotic 2 protons it can donate, and triprotic acids donate 3 protons. And polyprotic acids have multiple dissociation constants. So for each donation of for each exchange of a hydronium ion, there is a particular dissociation constant. So for the first one we have Ka1, then we have Ka2, Ka3, and in this manner it goes depending on how many protons it is capable of donating or taking up as well. Now, for example, we have the sulfuric acid that has that's a diprotic acid, and we have the sulfurous acid that is also a diprotic acid. We have the carbonic acid as a diprotic acid, and we have the orthophosphoric acid that is H3PO4, that's a triprotic acid, and when it deprotonates, we get different concentrations of H2PO4 minus, HPO4 minus, and finally PO4 3 minus. So it has three equivalence points, K1, K2, and K3 for the three protons that it donates. So next is a pH meter. So we need a pH meter to measure the pH of a particular solution or a buffer that we are actually making, or we have to do an experiment with. So these are the components of a pH meter. We have a pH measuring electrode. So we have an electrode that measures the pH of the solution. We have a reference electrode that we will keep to measure the pH. We have a temperature sensor and an input meter. Okay, so the pH measuring electrode is a glass pH electrode that utilizes a thin glass membrane that responds to the change in the hydrogen ion activity when it comes to measure the pH. And it works by a precision system where we have a voltmeter in the probe that measures the difference between the voltages of the two electrodes, and the meter then translates it into a pH value that we get and in the screen. Okay, so in this manner the pH meter basically works. So what we need to keep in mind when we are working with a pH meter is that the glass electrode so these are the factors basically leading to improper pH measurements, like when we do not store the glass electrode properly, if the glass electrode has some chemicals or salt deposition on it, that also the aging of that particular glass electrode, so there is a coating or clogging. Okay, so what we need to do is that before we take a pH measurement, we need to standardize the pH meter using different solutions that we have, like we have a pH a solution of pH 4, 7, 10, and we need to first standardize the pH meter, that is getting the instrument accustomed to the solution conditions, and also that we call as standardization. And when we standardize an instrument, it means we use the specific standard to calibrate an instrument, and for that we need reference pH solution. So a pH is known of a particular solution, we use them as the reference pH solutions. And why is this particular pH thing important in biology? Because most biological molecules are very sensitive to pH, because they have in them moieties, like you have the amino acids, nucleic acids that contain your bases, like the nitrogen base, the nitrogenous base, the hydrogen bonds, the sugar phosphate backbone, and these are very sensitive to any change in the particular pH. Even in the later weeks, we would be speaking about proteins, amino acids, and we will understand how sensitive a particular amino acid will be at different pH. Some would be in zwitterionic form, some would have a positive charge, some would get negative even at a minute change in a particular pH. So pH is very important in when we are working with biological macromolecules, and even pH affects the solubility of many biological substances, and also it hampers the structural and functionality of different proteins, mostly proteins, like we have the enzymes and etc. Okay, so when we speak of pH, we have to definitely speak of about buffers. So because it's all about the buffers that we need to work with when we are working with the biological solutions, the biological systems. So biological systems use buffers to maintain the pH. So what do we call as a buffer? So a buffer is a solution that resists to small changes in pH when we are adding either an acid or a base or even water. Okay, so that is resistant to a small change in the pH. We call that as a buffer. And chemically speaking, a buffer is a mixture of a weak acid and its conjugate base. So from the diagram, if we see that we have a okay. So from this diagram or from the picture, if we see that here is a solution of pH 7, okay, say water of pH which is neutral, a pure water of pH 7, and we add HCl and we add NaOH. The moment we add HCl, we see that the pH drops down to 4, and when we add NaOH, it becomes basic and it rises up to 10.5. But what is an ideal buffer? It will not change with a small amount of addition of acid or a base. So we have a buffer over here, and when we add an acid, it just changes by 0.1, and when we add base, it changes by 0.1. So the change is very small for an acid. So for example, we have the bicarbonate buffer, which is a mixture of a weak acid, that is the carbonic acid, and the conjugate base, that is the bicarbonate ion. And all the hydroxide and the hydronium ions added to the buffer are consumed in the overall steps, and but so it is consumed in the overall solution, but it does not change the pH to a greater extent. For a weak acid conjugate base pair, the buffering range is pKa plus minus 1. So that indicates that the change is small. Now this equation is very important, the Henderson-Haselbach equation. There are going to be many questions from this portion, and different tricky questions would be asked in exams as well as your assignments. So if someone has solved the assignment, they can understand that this portion is very, very important. Now, we have already spoken about the dissociation of weak acid, that is Ka equals to the product of the two products that we get from the weak acid dissociation and divided by the weak acid concentration of the weak acid. And when we take log on both the sides, we get minus log of H plus minus log of Ka plus log of conjugate base by acid. And this is pH, this is pKa. We have seen previously that minus log to the base 10 of a particular concentration is P of that particular thing. So pH equals to pKa plus log of conjugate acid, a conjugate base by conjugate acid. And so we can write this equation in this manner, where pH equals to pKa plus log of conjugate base by weak acid. So this is our Henderson-Haselbach equation. This is the derivation. And when pH equals to pKa, that is when the concentration of the conjugate base is equal to that of the weak acid, and that indicates the equilibrium situation. When the substrate is in equilibrium with the product formation, at that point the pH equals to pKa, and the concentration of the product as well as the reactant or the substrate is same. So there are a few applications of Henderson-Haselbach equation, because there is always a question that rises: why do we need to study this Henderson-Haselbach equation, or why is this even important? So because it will help us to calculate the ratio of the conjugate base to the weak acid if the pH is known to us, because pKa is a constant value. And if it will also help to calculate the pH if the ratio between the two concentrations are known, and it will help us to calculate the pH of a weak acidic solution of a known concentration. It determines the pKa of a weak acid conjugate base pair. Okay, and calculation of the change in pH when a strong base is added to a solution of weak acid, that can also be calculated, and we can represent that in a titration curve. I would be coming to all of these, means all of these points, mostly the second last point would be discussed in the next class probably, where I would be talking about the amino acids and the titration curve. So what is this titration curve? We would be coming in the next week. And also we would be calculating, we would be seeing how to calculate this isoelectric point or the pi. Okay, so these are the few of the several applications and the important applications of this HH or Henderson-Haselbach's equation. Now, when we have spoken about buffers, we also need to speak about few important or regularly used buffers in the field of biochemistry or biotechnology. Because these buffers, why do we use these buffers? Because these buffers have a particular range of pH which we can solubilize our protein in, such that we can carry out several experiments. The first one being that we would be studying about is the phosphate buffer. It mostly varies between a pH of 5.5, say 6.5 to about 8.4, 8.5, like that. So it has a very high buffering capacity. It is highly soluble in water. And the disadvantages is that, so okay, what is this buffering capacity? Buffering capacity is the ability of a particular buffer to retain its pH when strong acids or bases are added in the medium. In other words, it is the amount of H plus or OH minus ions that can be neutralized by the buffer. So higher the ability of the buffer to restrain itself from changing its pH, the better buffer it is in the case of biological molecules. And also we call it better buffer than any others. But this buffer also has a few disadvantages. That is, it inhibits some enzymatic procedures and reactions. It precipitates in alcohol, so the phosphate buffer cannot be used to precipitate your nucleic acids like DNA and RNA. Also, there is a possibility of this interaction with magnesium and calcium ions, and it interferes with different biological reactions. If I mention specifically, it is about the translation. It hampers the translation machinery of your um, translation of different proteins. So there is a possibility of that. The next one is stress buffer. It is also physiologically inert in nature. It is highly soluble in water, and it is also inexpensive. So these are the advantages. The disadvantages includes that the pKa of this buffer is about 8.1, so it has a very low buffering capacity. In the physiological range of 7 to 7.5, it is temperature sensitive, and sometimes it reacts with the normal pH electrode. So special electrodes are sometimes required. Now, the zwitterionic buffer, or we also call the goods buffer. So we have the mess. So these are all of the goods buffer that are basically used. We have the pKa at 20 degrees centigrade that is given. We have the pH range of these particular buffers. So goods is the name of a scientist, and we call the zwitterionic buffers as the goods buffer. So mainly telling about a few of them, we have the mess, we have the mops, the tris, the chess, and the taps. Okay, so these are the buffers, or also we call as the zwitterionic buffer or the goods buffer. So here are a few methods for buffer preparation, where we have the titration method, where the acids and bases are mixed and the pH is checked till we get a particular value. The other thing could be following the Henderson-Haselbach's equation. We know the specific ratios, or in which ratio we are going to add it. We add them, and then we check the pH in a pH meter, and we see that because it's following a particular equation, so we know the pKa, we know how much, what pH do we want at the end of the day, and then we add the ratio in that particular manner and we get it. At the same time, we have a few published tables where the recipe buffer solutions preparation methods are given. So we need to add, those are the optimized methods. So we need to add that particular salt or the acid to the total volume, and we get that particular pH. So the first one is the titration method, the second one is the manual Henderson-Haselbach equation method, and the third one is literature survey or the published tables. Okay, so mostly I would be speaking about the titration method, and so in this particular method we have two components. One is the acid and the other one is the base. So one is gradually added from the burette and the other is there in the conical flask. So this can be either the acid or the base, and here we have the other one, and we open the stopcock and the solution just drops, and we titrate it or shake it and mix it, and then we check the pH. That can be either checked using an indicator, like we have the phenolphthalein, we have the methyl red and methyl orange, we have all of these. Okay, or otherwise we can check it using the pH meter. Now, for example, for preparing a phosphate buffer, the monobasic sodium phosphate is added to the dibasic sodium phosphate dropwise till we get the pH. Now, what is the advantages of this particular method is that it's very easy to understand, usually useful when there is only one form of buffer that is available. And disadvantages is that it requires a lot of salt or base or an acid that requires mostly for most of the buffers, and it's relatively slow, and the variations in pH may not be considered. Okay, so the pH variation sometimes it's difficult to catch up because it's a slow process. Okay, so it sometimes takes some time. Okay, so I think that's all I have from the brief discussion about the lecture slide. So if you have any questions, you can ask me. I would be happy to answer them, and then we can go forward with discussing the questions and answers. If you have any questions, you can ask me about how much I have taught. And about the when I'm solving the question and answers, you people would be uh, solving from your side. Okay, and you can drop the options in the chat box. I would be checking them. Okay, and then I would be surely discussing with you the answers. But I want all of you to just try solving those questions and then doing it. Okay, now if you have any questions, you can ask me about the lecture slide. No questions? So everything is clear, I guess. Okay.
Hello, sir.
Yeah.
Uh, I have one question regarding to that. Uh, how to solve the PKB? How to get the value of PKB?
How to get the value of pKb?
Uh,
Okay, so to calculate pKb, so you see that for pKa, if I go back to my slide,
uh, your slide is not seen properly, sir. Hello, sir. Hello.
yeah, just a minute, right. Okay, right. Okay. see. So you know that pKa. So what is Ka? It is H plus into the concentration of the hydronium ion into the conjugate base divided by the weak acid. Now to get the pKb, it is from the perspective of the hydroxide ion. Okay, so for that your solution, for that you need to have like BOH is
uh,
equal to B plus and OH minus. So pKb or Kb is equal to your hydroxide ion concentration into your the conjugate acid concentration divided by the weak base.
uh, One more thing is here, sir.
Okay,
Even to buffers, preparation of buffer,
right.
some it means that yeah, some amount of ml given, some amount of ml given, and smaller value also given. So here it may be ammonium hydroxide and
I did not
another is
get you.
the
Sorry, I did not get your question. Can you just tell your question once more?
answer.
Yeah.
Yeah, but yeah, have value. Yeah. So suppose 60 some amount of ml of 0.2 mNaOH NH4OH given and some amount of ml of 2.0 0.2 m is given of NH4Cl. So how can we calculate the pH of that buffer solution?
So NH4Cl is given, NH4OH is given.
Huh.
Okay, so for that, okay, I would be discussing the question and answers. I think you will be getting your answer in that one only.
Okay, sir. Thank you.
So let us wait for 2 minutes. Let us take a break of 2 minutes and then we can start with the questions.
Okay, sir. Thank you.
Okay. So let us try solving the questions. Okay. So the first question is, a digital pH meter contains the following parts. So which of the following does it pH meter contain? Is it a glass electrode and monochromator? Is it a rubber electrode and voltmeter, glass electrode, temperature sensor, xenon lamp, and plastic electrode? So which of the following does the pH meter contain? So I have mentioned in my slide, it is the glass electrode and the temperature sensor. That is the correct option. So option C. The next one is the concentration of H plus ion in 1 millimolar NaOH is. So what is the concentration of H plus ion in 1 millimolar NaOH? So for that we need to get back to our equation, that is concentration of OH minus is equal to 10 to the power minus 3. 10 to the power minus 3 molar, because it is given in millimolar. So 10 to the power minus 3 molar. So, and then we have Kw is equal to minus. So ultimately this is 10 to the power minus 14 is equal to 10 to the power minus 3 into so then from here you get the value to be A. Sorry, this is 10 to the power minus 11 molar. Yes, so bold. So D is equal to 10 to the power minus 11 molar. So that will be the concentration of H plus ion in, okay, so this was, sorry, this is H plus. So concentration of H plus ion in your 1 millimolar of NaOH. Okay. The next one. Which of the following sentence or sentences is or are true? Buffers are made up of weak acid and conjugate base of another acid. If pKb of a base increases, it becomes weaker base. For an acid, if temperature increases, then the pH will be decreased. For a 10 ml buffer solution, if we add 5 ml of distilled water, then the pH of the buffer gets changed. So let us see the option one by one. Buffers are made up of weak acid and conjugate base of another acid. Of another acid is incorrect. It has to be of a weak acid and the conjugate base of that same acid. So this one is incorrect. Number 2 is if pKb of a base increases, then it becomes a weaker base. That is correct. So 2 is correct. And for an acid, if temperature increases, the pH gets decreased. That is also correct because pH is temperature dependent. And when the temperature for an acid, if the temperature decreases, the pH gets increased. Vice versa, for a base, if the temperature increases, the pH gets increased. And for a base, if the temperature decreases, the pH also gets decreased. So this is also correct. For 10 ml buffer solution, if we add 5 ml distilled water, the pH gets changed. That is wrong because a buffer is a solution that does not change on the addition of small amount of acid, base or water. Now getting to the next question. A researcher accidentally puts 1.6 gram of NaOH pellet in 1 liter 0.5 molar acetate buffer of pH 4.74. Now what would be the change in pH? Ka equals to 1.8 into 10 to the power minus 5 for acetic acid. Okay, we will come back to this question in the last because I need to show the calculation. The next one is, what would be the optimum pH range for the Hippis buffer of pH 7.6? So if you can remember from the zwitterionic or the goods buffer where I showed, so the optimum pH for a Hippis buffer is 6.6 to 8.6. The next one is, which of the following is or are true? MOPS and MES buffers are goods buffer. TRIS is a zwitterionic buffer. Phosphate buffer may inhibit an enzyme activity. TRIS buffer closely resembles the intracellular environment. So TRIS buffer resembles an intracellular environment, that is wrong. Whereas on the other hand, what we have is MOPS and MES are goods buffer. TRIS is a zwitterionic buffer or a goods buffer, and phosphate buffer may inhibit an enzyme's activity. So all of these three is correct except 4. So this is the correct option. Now here the Ka value is given. We need to find out the Kb value. So how do we do it? So we know pKa equals to minus log of Ka, and from there we calculate the pKa value, and then pKa plus pKb equals to 14. I've shown that. So therefore pKb equals to 14 minus the value that we get. So if we calculate the pKa, it is coming about 5.25. That is the value of pKa. To get the pKb, it is 14 minus 5.25. That will be about 8.75. Okay. So B is the correct option. I am repeating once more. Minus log of Ka equals to pKa. So when you do minus log of this, you get 5.25, and pKa plus pKb equals to 14. So pKb equals to 14 minus 5.25, that is 8.75. Now the next question is, which of the following is or are goods buffer? So it's a similar kind of a question. We have the Hippis, MOPS and MES. Phosphate is not a goods buffer. So 1, 2 and 3 are correct. These are some direct questions. Okay. Now pure water is neutral. Why? Because it has pH 7 in experimental conditions, equal amount of H plus and OH minus ions. Water does not contain free H plus and OH minus ions, and water cannot ionize. So pure water is neutral because it has, from the calculation or experimental perspective, it is definitely neutral because it has a pH 7. That is true, but the more accurate option would be it has equal amounts of H plus and OH minus. Okay. Buffers with high buffer capacity. What does that mean? So buffers with high buffering capacity. So as I mentioned, buffer capacity is the ability of a buffer to retain its pH when strong acids or bases are added. That means if you add higher concentration of acids or base, only then it is possible to change the particular pH of that buffer. Otherwise, the buffer remains unchanged. That is meant by buffers with high buffer capacity or high buffering capacity. Now here we have another calculation. So these calculations I would be showing later. Let's go directly. First, let's do the direct questions. Okay. So buffers used for biological experiments should have the following characteristic: chemically stable and water soluble, should not interact with biological molecules, can change pH easily, should have high ionic strength. So buffers that we use for biological experiments should not change pH easily and should not have a very high ionic strength, but it should be chemically stable and water soluble, and also it should not interact with the biological molecules. So therefore 1 and 2 are the correct options. Now the next question is, here are 4 sets of solution of different pKa values: A, B, C, D, pKa 3.5, 5.5, 8.5 and 12. Which of the following statement is or are true? Acid A dissociates to a greater extent in water than acid B. C and D are basic in nature, whereas A and B are acidic in nature. D is the conjugate base of C, and acid A is more likely to be polyprotic than acid B. Now, from the compounds, we cannot, just from the name of the compounds, we cannot tell whether it is a polyprotic or a diprotic or a monoprotic acid. So D is incorrect. D is a conjugate of C, that is also we cannot tell. What we can tell is that acid A will be dissociating to a greater extent than acid B because it's a strong acid and this is a weak acid. Whereas C and D is basic in nature and A and B is acidic in nature. So this is a strong acid, weak acids, weak base and the strong base. So A and B are the correct options. Okay, the next one. Which of the following is true? Tap water is used in biological experiment. Thermal shaker can control temperature and rotation speed. 100% ethanol is used for surface sterilization, and thermal shaker and cooling centrifuge are the same experiment. Same equipment. So the first one, tap water is used in biological experiment, is absolutely incorrect because we use milky water, and that is also two types, type 1 and type 2. One is DNS free and 2 is DNS and RNS free. The type 2 water is DNS free and type 1 water is DNS and RNS free. So tap water cannot be used. 100% ethanol is used for surface sterilization is incorrect. We use 70% ethanol. Thermal shaker and cooling centrifuge are the same equipment. That is incorrect. They are two different equipments. Thermal shaker can be used to control temperature and rotation speed. That is correct. Okay. Buffers are a mixture of, direct question, it's a mixture of a weak acid and their conjugate base. Okay. The next question is, okay. Two researchers have 5 sets of buffer 7.4. The pH was measured in pH meter. So set A 7.29, 7.4, set B 7.4, 7.1, set C 7.39, 7.39, and in set D you have 7.35 and 7.4. So who is more accurate and who is more precise? The question is that. So you know the particular pH buffer, so it was measured, so 7.29, 7.4, 7.39 and 7.35, and here we have 7.4, this and that. So for this we need to do some calculations, and as per our calculations it says that, so here we need to calculate the average of the points and the standard deviation. So the average and the standard deviation of the values measured by A is 7.38, and the standard deviation is 0.054, while that for the values measured by B is 7.4 is the average and 0.086 is the standard deviation. So the average value of B is closer than the actual value to the actual value, that is 7.4. So B is more accurate. And also the measurements by B are grouped more tightly, that is resulting in smaller standard deviation. Hence B is also more precise. So BA is less accurate and precise than B. So D is the correct option. So buffers used in biological experiments, so this I discussed, and buffer should be a mixture of was also being discussed. So okay, now let's get back to the two questions that we skipped. Okay. Okay, the first question was, a researcher accidentally puts 1.6 gram of NaOH pellet in 1 liter of 1 liter 0.5 molar acetic acid acetate buffer of pH 4.74. Now what should be the change in pH? So how you do 40 gram of NaOH equals to 1 mole and 1.6 gram NaOH equals to 0.04 moles. Now NaOH in solution is 0.04 mole divided by 1 liter, so you get in molar, 0.04 molar. And now when you go on to calculate it, so the initial you have 0.5 molar and this will also be 0.5 molar, and for OH equals to 0.04. So in equilibrium condition you have 0.46 molar and 0.54 molar. Now so the change, to get the change, it should be, because from here the change is getting negative and here it's the product side, so it is getting positive. So 0.46 minus x and 0.54 plus x, and here you have x. So when you calculate the Ka, it is 0.54 plus x into x divided by 0.46 minus x. And so x equals to, when you calculate it, x equals to 1.53 into 10 to the power minus 5. So pH minus log of that value, that is 4.82. And what is the change in pH? So the change in pH is 4.82 minus this value, that is 4.74. Okay, so 4.82 minus 4.74, that is 0.08. So the change in pH. So how I did it? So the first one, 40 grams, that is the molar weight of NaOH, that is 1 mole. So 1.6 gram of NaOH, that is 0.04 moles. Now from moles I calculated the molarity divided by 1 liter, so 0.04 molar. And in the primaries, in the first stage you have 0.5 molar, 0.5 molar, and on addition of 0.04 molar you get 0.46 and 0.54. Okay, and then what would be the change? That is minus x and plus x plus x. So now putting it into the Henderson-Haselbach equation, 0.54 plus x into x divided by 0.46 minus x, and there you get the value of x and you can calculate it. Okay, and the other question was okay, yes, a buffer is prepared from 65 ml of 0.2 molar NH4OH and 35 ml of 0.2 molar NH4Cl. Okay, I think that was the question that you were asking me. So this is the buffer is prepared from 65 ml of this NaOH and 35 ml of 0.2 molar NH4Cl. So what is the pH of the buffer solution? So the Kb is given. Now you can calculate the pKb also, and so if the two solutions are mixed, they both have been diluted. So 65 plus 35 ml is 100 ml, and NH3 final equals to your you have 65 ml into 0.2 molar divided by 100 ml, that is you get in molarity. Okay, so you get in molarity. So 65 ml into 0.2 ml divided by 100 millimolar, it is 0.13 molar. And you do the similar thing for NH4 plus, because here you have NH4. Okay, so this is NH3 also, you can tell, because it is NH3 plus H2O. So let's think it of as an ammonia. So you do it in that manner, and this is NH4 plus. So you have 35 ml into 0.2 divided by that. So it is 0.07 molar. So initially you have 0.13 on adding, because it has decreased, and this has also decreased from 0.2 molar, and here the initial concentration is 0.07. So what is the change? Again in the same manner, minus x plus x plus x, and you get the equilibrium. Now pKb equals to your x into 0.07 plus x divided by 0.13 minus x. So ultimately what you get is x equals to 3.342 into 10 to the power minus 5. So POH. So here we have been asked to calculate the what is the pH. So POH, when you calculate, you get 4.48. 14 minus 4.48 equals to 9.52. So you get the pH to be 9.52. And in this way you can calculate it. So first you calculate the initial concentrations on adding, because they get diluted. Second, you calculate the change. To calculate the change, you have to take a particular value, say x, and for the reactants we have minus x, for the products we have plus x, and then in the equilibrium state it is the final equilibrium concentrations, and then you put it in the particular equation of pKb, and you get the POH. When you get the POH, you can calculate the pH. In this manner you can do it. Okay, I think it's clear to you. All right.
Sir. Okay.
Okay. Okay, that is great. Okay, so that's all I have from my portion. Uh, and then if you have any question, you can ask, or else we can end the class. No more questions?
Sir, no.
Okay. Thank you then for joining this session. Again, we will meet on next Monday, same time, 6 to 8 p.m. And we will
Ah.
be discussing with the week 2 lectures. Okay. Thank you. Thank you all.
Tomorrow have a same time lecture.
Sorry?
Hello, sir.
Yes.
Tomorrow have same time lecture.
Tomorrow, actually, this same thing would be taught to you. The same things would be
Uh-huh.
discussed, but by a different teacher.
Acha, okay.
So, you means it will be two live sessions. More or less, the
Uh-huh.
same things would be taught. Means any class you are
Uh-huh.
comfortable in joining, you can join.
Okay.
Either Monday or Tuesday, 6 to 8. Okay, good. Thank you.